Okay, welcome everybody to this ERN-EYE webinar. And it's going to be a very interesting webinar on CRISPR and antisense oligonucleotide RNA-based therapies. And as you probably, many of you know, the gold standard, if you can speak already of a gold standard, I would say, in treatment of inherited retinal diseases, has been gene replacement therapy using AAV vectors. But for hundreds of genes involved in IRDs, they are still not treatable. And some of those involved genes are extremely rare. And some of the common genes, think about USH2A, for example, are much too large to fit in this AAV vector and do this gene replacement therapy. So we need new approaches to try to tackle as many of those blinding diseases as possible. And key new techniques that are being used are antisense oligonucleotide RNA-based therapies and CRISPR-based therapies, but there are a lot of hurdles still to take. And in this webinar, it is a pleasure to moderate three world-leading experts in the field who are going to talk about these topics. And I would like to immediately start with the first expert on the topic of CRISPR-based therapies, who's going to be talking about this topic, is Dr. Pietro de Angeli from the University Hospital in Tübingen and until quite recently also Columbia University. And he's going to talk about therapeutic promise of the 21st century, strength and limitations of gene editing. Dr. De Angeli, the floor is yours.
Thank you. Good afternoon, everybody. I hope you can hear me well. So for today's presentation, I thought of preparing a more general presentation where I would highlight the power and limits of genome editing without going into much detail so that a broader audience can also follow. So before starting, I would like to define what genome editing is and why it's relevant for our field or for the field in general. So by genome editing, we mean means by which DNA can be precisely changed. And we refer to that as also targeted genome editing. And why is this relevant? Because as mentioned already, if we look at inherited disorders, and in our field specifically inherited retinal disorders, which are caused by mutations, these mutations, which are changes into the code of a gene, can be reverted back to wild type by implementing genome editing technology. So arguably, the patient could be treated by this technology. If you look at the different genome editing tools that are available to us, we see already that 30 years ago, the first genome editing tool was discovered, and these were zinc finger nucleases, which together the TALEN nucleases were the first ones who enabled genome editing. However, the implementation of these tools was really complex and laborious, and they received little translational upside. For this complexity. Then in 2012, when the CRISPR-Cas9 gene editing tool was discovered as a tool able to edit the DNA by two scientists, Jennifer Doudna and Charles Tentier, this received immediately a lot of interest because of its versatility and ability to be reprogrammed in a really easy way. And today's talk will focus on CRISPR-Cas9 for this reason, because it is the tool that started the genome editing revolution. Going to the next slide. So looking into the power of genome editing, specifically focusing on CRISPR-Cas9, we'll have a look at molecularly what this tool enzyme does. And molecularly, it is an RNA-guided endonuclease composed of two different elements. We have a Cas9 here on the left side, which is the endonuclease. So it's the enzyme that is able to generate double-stranded DNA break, which complexes to a guide RNA. And as the name suggests, the guide RNA is guiding the specificity of the enzyme towards a specific sequence of our interest. So these two elements complex together, and they can scan through whatever DNA to look for full complementarity of the guide RNA to the target sequence. And when this full complementarity is found, the Cas9, which is again the enzyme able to generate a double-strand break, can introduce a double-strand break. So by simply changing the sequence of the guide RNA, you can immediately understand that any sequence virtually across the genome can be targeted and being accessible for editing. So what happens once a double-strand break is generated? The cells sense this as a damage, because double-strand breaks are never good. So they start the repair mechanism of the double-strand break. And these repair mechanisms are mostly 3 of them. We have on the left side non-homologous enjoining and microhomology on mediated repair, which are error-prone repair pathways. What does it mean? It means that the break is resolved, is repaired, but at the targeted site, you have the insertion or deletion of sequences. So the resulting sequence will not be as the initial one. And why is this important? If you think about toxic proteins, for example, and they become toxic when there is a mutation. I'm just generalizing the issue here. If you can switch off the expression of this toxic protein, of course, you can achieve a therapeutic benefit. And in our field, could be the example of rhodopsin-linked retinitis pigmentosa, where the pathogenic variants are acting through a dominant negative effect. So if you can switch off the mutant allele specifically, you can arguably achieve a therapeutic benefit. And then on the right side, you have HDR, which stands for homologous directive repair. And this is a repair pathway that leverages on a donor template that you see down here. So a donor template is a sequence of DNA that we can provide the cells with. And if this donor template is designed properly, meaning that it has homology arm to the double-strand breaks, it can integrate exactly at the double-strand breaks. And if the donor contains the sequence, wild-type sequence, of course, this can revert the mutant sequence. So if you think about implementing these repair pathway, you can understand that potentially every mutation could be targeted. However, then reality comes into play, and the question is HDR really the holy grail? It's a rhetorical question because the answer is no. And why is that? So homologous directive repair is only active in mitotically active cells. What does it mean? Those cells that are actively proliferating, proliferating. And examples are stem cells, blood cells, and liver cells. But we are interested in photoreceptors. When we look at photoreceptors, of course, they are post-mitotic cells, and HDR is not practicable. So you already see that this tool that could potentially enable targeted genome editing specifically down to the base pair is not practicable for the disease of our interest. So what's the solution here? Scientists came up with a more advanced genome editing tool, and there are 2 actually. So I will present to you the first one, which is base editing, and then we'll go to the second one. So base editing is based on CRISPR-Cas9 ability to specifically look for a sequence. So what happens is that if you fuse Cas9, which is depicted here by this L shape, to a deaminase, which is an enzyme able to deaminate specific base pairs, what you can do is that you can direct the activity of the deaminase towards a specific sequence. And we have 2 types of deaminases, cytidine and adenosine. The cytidine would deaminase cytosines to uracil, and this would be recognized by the cells as T. So by deaminating a C, you can generate a C to T conversion. As you can see here, CBE, cytosine base editors, can make C to T changes, and adenosine base editors can make A to G changes. So basically, we can do transitions with these tools. And when we look at the human genetic variations or variants, associated with disorders, you see that around 30% of variants available on ClinVar are transition point mutations. So by implementing base editing, one could think of targeting 30% of all the mutational spectrum across disorders. But what about the remaining 70%? So scientists didn't stop there, went on and came up with prime editors, which is also referred to as the all-doing editing enzyme. So again, we have Cas9. In this case, Cas9 is fused to a reverse transcriptase. A reverse transcriptase is an enzyme that is able to convert RNA to DNA. So what it can do through the specific guide RNA that is guiding the specificity and specifying for the sequence to be written directly into the target DNA, what the retrotranscriptase does is exactly to retrotranscribe the sequence template that we give the system into the target DNA. So by using this enzyme, we can mediate all insertions, deletions, and all 12 possible base-to-base conversion. So technically and molecularly, we see that the available CRISPR-Cas9 based editing tools are really powerful, versatile, and can, again, molecularly target any mutation across the genome, right? But then what is the next step? We have, of course, the engine, but how do we deliver the editing tools, for example, into the cells? And here some limitations come into play. So I define two main limitations because, of course, it's a complex issue. So technical limitations and translational limitations is in the plural form, but for the technical limitations, I would like to focus on the delivery issues. So we have our editing system when we have to deliver that to the tissue of our interest. And if we are going after metabolic disorders, the liver could be the target. If we go after inherited retinal disorders, of course, the eyes would be the target. And as for now, we don't have a single modular platform that enables delivery across specifically across the different organs. And if when we look at the considerations that are behind designing a proper delivery method or platform, we come across tissue specificity, because as said, you want to be specific for the tissue that you want to target and not thinking of targeting the brain, and then all the editing would also happen in the other cells across the body. And this is especially relevant when you think about the possibility to get editing into the reproductive systems, which then would be transmitted to the offspring. Then another topic that is important is the editing threshold. So how many cells one need to edit to have a clinically meaningful benefit, and then immunogenicity. So when we look specifically for the eyes and the different protocols for delivering gene editing tools into the eyes, these are mostly subretinal or intravitreal. And the eyes as an organ is having different advantages for the delivery. So it's immunoprivileged. The accessibility is easy, of course, because it's outside. And then also the dose required for treating is lower compared to other organs. You can easily monitor the clinical benefit through different functional assays, and you have a bilateral control because you can treat one and you have an internal building bilateral control. I put it in brackets because there would be more considerations to be done there. However, if we look at the gold standard today for delivering transgenes, including CRISPR-Cas9 into the eyes, the gold standard is AAV, so adeno-associated virus. So these are great delivery systems for gene augmentation where you need the persistent expression of a transgene. However, for the gene editing, you just need a hit-and-go situation. So the prolonged expression in post-mitotic retinal cells is an issue. And this is an issue because it can induce more off-target. And when I say off-target, it means that CRISPR-Cas9 is specified for a specific sequence, but of course it can get confused if there are sequences that are really similar. So it can also induce editing in these sequences, and the longer the worse in terms of targeting that one can get. And this goes along accumulation of genetic translocations, for example, or genotoxicity, and also immunoresponse. The more this transgene expressed, and the more chance of immunoresponse one has. Really quickly, going into the translational limitation, which I think is really interesting and important to be aware of. So basically, it's really too expensive for being sustainable for rare and ultra rare and N of 1 variants. So if you think about going into treating the patients, this takes at least 3 years and 5 million to get from the bench to the bedside process, so that the FDA can clear your approval investigational new drug. And this is often a financial limitation, especially for rare patient populations, because 5 million is really a lot if you think that there is just one patient to be treated. And of course, also the time is a limitation when thinking about really devastating metabolic disorders where children only have 8 months to live, because there is the ability and a possibility to treat these patients, but then the reality comes into place. However, on the bright side, there are initiatives trying to push into the direction of more accessible treatments for CRISPR-Cas9. And one example is from last May when KJ was treated for a metabolic disorder, and what they did went from the genetic diagnosis to the treatment in only 7 months, showing that the time constraint can be really squeezed. However, the cost, of course, they didn't care at that stage because it was an academic endeavor. Solutions. So this is my last slide about solutions, how we can try a framework to go quicker into gene editing approaches into the patient. So if one has modular development and the risking strategies, and FDA would open up to regulatory pathways for personalized therapies, then of course this would enable easier access for patients. And there is now a formalizing of this strategy into a company, for example, Aurora Therapeutics, and you can have a look more there, how they are developing to try to tackle these issues so that more patients can get access to therapies. So really briefly, conclusions, we saw that we have available gene editing tools that are molecularly powerful and versatile. Comparably, the development of delivery tools effectively supporting in vivo delivery are somehow falling behind, and we need a paradigm shift in the regulatory framework and clinical trials designed to hope to scale these strategies up. Yeah, that's it from my side.
Thank you very much, Dr. Angeli. It was a wonderful overview, also covering the challenges and the possible translational opportunities, which are quite complicated. I guess we will come back to that at the discussion at the end. For now, I would like to proceed to Professor Mieke van Halest from Amsterdam UMC in the Netherlands, who's going to talk about her initiative of the CRISPR Clinic.
Thank you very much, Camiel and the organizers for letting me presenting here the initiative of our CRISPR Clinic in the Netherlands. And I'm really pleased that Pietro already discussed all the technical parts, because as a clinical geneticist, as a clinician, I'm presenting this initiative. As a clinical geneticist, we realize that we see patients, we do diagnostic analysis, and we identify the cause of certain disorders. But then what can we do? We can help them by explaining what the disease is, what the future might bring, what the risk is for SIPs, but also future recurrence risk for the next generation. But I am always a bit frustrated that that's it. I prefer to prescribe medication or to offer treatments. And that's why I'm very pleased that 2 years ago, I decided with my colleagues to organize a CRISPR clinic for patients who have questions about all the news and the promises that are in the media. The CRISPR clinic is part of see if I go to the next one, part of the Department of Human Genetics. That's where my clinical genetics department is as well, the clinics, and also EMA Center for Personalized Medicine. The EMA Center for Personalized Medicine is new. 4 years ago, I established this center together with my colleague Clara van Karnebeek, because from her part, from the pediatric department, and I'm from the human genetics, we got the initiative to make sure that patients will have many unmet medical needs and opportunities, and we would like to get more knowledge and provide more care for those individuals with suspected rare genetic disorders. And we work according this P4 medicine in a multidisciplinary centers. You see, personalized, the patient is central. We want to offer predictive, preventive options, but also the participation of patients and clinicians, and building an infrastructure and uniting those expertises. Because I realize as a clinical geneticist, you might have an interest in disease A, and maybe somewhere else in the big academic centers, there might be some other research groups working on the same disorder without even knowing that it might help each other. So the new infrastructure is important. And we will thus organize a central point in our academic center here in Amsterdam to accelerate, but also to innovate personal treatments for those cases with these rare genetic disorders. We focus on 4 diseases. Eye is important, so the eyes, working closely together with the ophthalmologist like Camiel, working closely with the metabolic people. Clara van Karnebeek has her specialization in that. Also with neurodevelopmental disorders, many syndromic intellectual deficit cases, and also food satiety disorders. And the aim is to bring the translational results or the innovative results in the daily clinic, that the patients are well informed about future options for their disorder. So connecting, accelerating, and innovating. Patient central, this P4 as I mentioned, and we have set up since then many multidisciplinary and cross-departmental meetings, clinics for diagnostics, therapy, care, but also education is extremely important, because as we are sitting here together, we might know everything about these new options about CRISPR therapy, etc. But the colleagues who are already 20 years in their expertise, they might not have a clue, or they're not that interested, but also the future generation, they need to be up to date. So that's part of it as well. And we would like to have customized care for any patient that we see. So we are working according to this catalyst wheel. You see 3 different colors. The blue is the diagnostic part, the red is the preclinical part, and the orange part is the clinical care, and also getting therapy into the clinic, as we just heard, regulatory bodies, pathways, reimbursement, etc. And this pie is being served on a silver plate of ethics, training, valorization, and education, because that's all part of our center as well. We were lucky when we initiated this that the government, the Dutch government, provided 10 million euro to attract assistant professors. And in the end, we were happy that we could appoint 30 people, top talents, to work on all our projects. So one of the topics is, as I mentioned, eye disorders. So these 4 clinics working are meeting in separate groups, but the part that I'm presenting today, so the CRISPR clinic, is overlapping, because we just heard about baby J, who has been in the newspapers, and every day when such a new news item pops up, then here at the Department of Clinical Genetics, the telephone is ringing 24/7. Every patient who reads about this, every patient that's waiting for new treatment options, they want to know more. They want to know, hey, why is this possible for this child? Can you help me, please? So there's an unmet medical need that patients have questions. And we have initiated this CRISPR question and answer clinic, where scientists, so the basic scientists, clinicians, but also patients meet together. The patient can ask questions, and they often come to the clinic with bunches of scientific papers to inform us about their specific disorder. But they also need to understand more of what we just heard, the cost aspect, the safety, etc. It's not just going to the dentist and say, okay, my teeth is not well, just take it away and place a new one. So it's extremely important to communicate well with all the patients that have questions for that. So often it happens either with a news item a popular news item, or with some scientific papers that we published ourselves. This is a Dutch case of a Dutch manuscript you might not read and not be able to read. But after this, we got a message that somebody who read this paper was wondering if she, the lady herself, was a doctor. So those are the patients that read actually the scientific papers. They can understand it, and they were wondering if they could come to the CRISPR clinic because they really want to know if they could be a candidate for such a treatment. And those are the patients that come actually to our clinic. And this is Bas, the patient in the square in red. This is his pedigree. So he has 3 daughters, and Bas himself was diagnosed with, as you just read, Best disease, based on a pathogenic variant in the BEST1 gene. And unfortunately, even though he had a 50% chance of having a child with with this disorder, all 3 his daughters were affected. And he really wanted to know if, although he was already having problems with his vision, he could be in time to prevent that his daughters would turn blind as well. So just about the disease itself, the verrucoular macular dystrophy caused by the pathogenic BEST1 variants. So Bas, the father, and all 3 his daughters had this, unfortunately, this gene defect. And the ophthalmologist might all know, and also the patient might just know, it's a dominant disorder, meaning that it can pass from a generation to generation, 50% chance per for a new child. It encodes for the bestrophin 1, which has a channel function across the cell membranes in the retinal pigment epithelium. And in the pictures below, you see that there on the right side is central loss of vision and also the loss of the ability to perceive colors and details. And ophthalmologic investigations show the pictures as you see here. This is the normal one, and this is the patient with BEST1 gene variant. So with our CRISPR clinic team, my colleague Philip Janssen, he was sitting in that clinic as well. And coincidentally, he went to London just for a scientific meeting on any disorder. And then his eye caught, from the corner of his eye, he saw a poster. And when looking at that poster, he realized that there were actually some answers available for Bas and his family. So he saw this poster, and we also heard that in the previous talk, we need to collaborate to help the patient. So this group, Amanda Carr is a senior author, senior researcher. She was actually working on BEST1 to provide CRISPR gene therapy. So fibroblasts of the patients, iPSC were being generated, and then editing the gene defects. And this is what was on the poster. So quite exciting, just a week after our CRISPR clinic. By then, we had already seen, I think, 20 cases in our CRISPR clinic, and the majority of the cases are wondering about intellectual deficit or disorders that affect the brain. And we just heard the eye is a good target, as well as the liver. So this could be an option. So ever since then, we started a collaboration, and Amanda Kerr, the senior researcher, already successfully edited the iPSC lines from the BEST patient, and she had some positive effects at the moment. So exciting. But we realized that when we have this clinic, we also mentioned that we shouldn't overpromise, but also not underpromise. In the past, when patient came to my clinic for asking about future treatment options, I had always to disappoint them, to say, okay, it might take a few decades, but nowadays it might be sometimes under the 5 years. We just heard 5 million, 3 years, but the last newspapers were promising, as we just heard, 7 months. And that's the aim of our CRISPR clinic, and that's the aim of the human genetics, but also of the CRISPR, of the MS Center for Personalized Medicine. We want to make sure that we can start organizing all the different steps available and see which patients we can help. So the first step for this is that Bas and his family, they can have a skin biopsy. Bas can have a skin biopsy, and it can be in collaboration with the research group at UCL, checked for the iPSCs, and to see if it can be a target, if it's as successful as the previous results as well. So what does it mean for the affected family? So I'm just mentioning here how to inform, how to communicate with patient, not over-promising, because there is always hope. That's what you read in the email that Bas' wife sent us. The skin biopsy is important, but the most important question is, okay, if we can find a cause, a treatment for Bas, he's willing to have a treatment, even though he can't be helped because he already has vision loss. But we might be in time for his daughter. So clear communication and managing expectations is extremely important. So yesterday I spoke to the family. That's why they agreed to show the pictures of the family, and we will organize this skin biopsy and the cell lines to be sent to London. And well, the cliffhanger is that on the 9th of June, you are more than welcome to visit our Emma Center for Personalized Medicine Symposium at Amsterdam, because we will then further discuss the continuation of this story. And I really do hope that we can help these and other patients in the future with these new therapeutic options. So I would like to thank the patients and the families and give back the word to Camiel.
Thank you very much, Mieke, for this wonderful overview and a great initiative where all the disciplines come together. It has been a pleasure to be just a small part of such a puzzle that you are trying to solve. We're going to discuss it later on. I now give the word to Professor Collin from Radboud University Medical Center in Nijmegen, the Netherlands, who is a world-leading expert in the field of antisense oligonucleotide therapy. And just yesterday, I got an email that in Amsterdam, our first patient who was screened for USH2A exon 13 RNA therapy, antisense oligonucleotide therapy in the LUNA study was approved for inclusion. So this shows that things are going fast now and much more is to come. So looking forward to your talk. Thank you very much.
Thank you, Camiel, for the nice introduction, and great to hear the news about the patient inclusion, and that indeed the antisense oligonucleotides are moving ahead. Antisense oligonucleotides are the therapeutic molecules that we use, for instance, for splicing modulation therapy, and I will focus on that in my talk, and also share a little bit of our first ideas to really move towards a personalized intervention, meaning a single oligonucleotide for a single patient. So these are my disclosures, and I think I can skip the introduction pretty quickly. The only thing I want to stress here, on the left you see the eye, on the right you see a nice drawing of the retina, and it's a very complex system with many genes involved, many proteins involved, and in dysfunction of any of these genes that are expressed in these retinal cells, they can lead to a subform of retinal disease, and it's clinically heterogeneous, but also genetically heterogeneous, with several hundreds of different genes associated with IRD. And to make it more complex, you have these different phenotypes, these different genes, and it's also known that pathogenic variants in some genes, depending on which variant it is, can either cause a more cone-like phenotype or a more rod-like phenotype, an early onset, a later onset, and everything in between. What's important when we consider splicing modulation therapy is that 15 to 20% of all the different mutations or pathogenic variants, they affect the pre-mRNA splicing process. And for those who are not aware, pre-mRNA is an intermediate molecule between our core genomic DNA and the ultimate functional unit, the protein. So pre-mRNA is transcribed from a gene, and after that, the pre-mRNA splicing occurs. And in that step, different things can go wrong because of pathogenic variants. And these pathogenic variants, they are here indicated with the asterisk. Let me see if I can add the laser pointer. So these different black asterisks, these are pathogenic variants, and you will find them in exons and in introns. And altogether, they can have different effects on the pre-mRNA splicing process. The end result, however, often is that a protein that's synthesized, if it is synthesized, is not functioning. And if the protein doesn't work, the cell doesn't work, the system doesn't work, and in this case, the transmission of visual input to the brain is affected. So we came up already almost 20 years ago with a way to modulate that. And we want to use antisense oligonucleotides for that. It's a different therapeutic approach than the CRISPRs we've heard about. So here in green, you can see the antisense oligonucleotide. It's a small RNA molecule, a synthetic RNA molecule of between 17 and 22, 23 nucleotides. And it works as a molecular band-aid to block the machinery that does the splicing to recognize elements that should not be in your end product. So here again, you see the pathogenic variant. It leads to a PE, a pseudoaxon insertion, with the result of no protein. But if you mask the recognition of that pseudoaxon, then protein synthesis will occur properly because the mRNA splicing is occurring in the way that you want. Then we have, you need to chemically modify such an ASO. There are all kinds of ways of delivering it. We've done quite a big deal of work to find the best conditions, and I will not go into detail during this talk. So as said, I think ASOs are a little bit more advanced compared to CRISPRs. So the initial work for SEP290 and also for H2A, that's the study that Camiel just mentioned. We've done preclinical investigations at Radboud University, collaborated with a Leiden biotech company, ProQR Therapeutics, who drove these programs all the way to clinical testing. And I would highlight the first study just to shed a little bit of light on where we are and what the challenges are. So SEP290 is a protein expressed in the photoreceptor cell, and in particularly in the connecting cilium, which is basically the highway that transports proteins from the inner to the outer segments. And you can imagine that if such an important protein for transport is not working, that also the light conversion doesn't take properly place. So pathogenic variants in SEP290, they are very severe and they cause early onset Leber congenital amaurosis. So in 2006, my colleagues in Nijmegen, they discovered a deep intronic SEP290 variant, accounting for up to 15% of all LCA cases in the Western world. So that's quite a common variant. And this variant, basically, it creates a pseudoaxon here depicted in red. It's a similar mechanism to what I described with PE. This pseudoaxon insertion contains a stop codon, so the SEP290 protein is not properly synthesized. How does this ASO now work, this molecular band-aid? It's designed in such a way that exactly hybridizes to the region that you want to block. You saw these splicing factors coming in, but because of this ASO binding, they cannot find their target and they leave again. They are not able to recruit the right RNAs and proteins to properly splice that part in. And the result of that is that you get a correct mRNA and a correct protein. So it took us 10 years of preclinical research to further develop this from idea to really test that it works in a cellular model. And we also did attempts in an animal model. But in view of time, I will only describe the results of the clinical study, which was sponsored by ProQR Therapeutics. So it was a phase 1/2 clinical study where patients received unilateral intravitreal injections of the ASO. The advantage of an ASO is that it's way smaller than, for instance, the CRISPR machinery. So in terms of delivery, it's a little bit less complicated. You do need to dose it every 3 months because it affects the RNA, and the RNA, of course, it's not stable and doesn't live forever. And by the unilateral injection, you are allowed to compare a treated versus an untreated eye. And this is a picture of the best responder of that study. I think there were 10 people participating, and one was a super responder, that's this person. But you can clearly see here the triangles. This is the treated eye versus the crosses here, that's the untreated eye, that there is a significant improvement in both achromatic as well as chromatic acuity. So the phase 1/2 study was definitely considered a success. However, a couple of years ago, after the phase 2/3 study was started, we were confronted with the fact that in that study, the primary endpoints were not met, nor were notable secondary endpoints. Later on, a post hoc analysis still demonstrated encouraging signals of improvement, but overall the trial failed. So the first thing, of course, you then go and do is compare these two different setups of these studies. And where initially there was a relatively high dose used, an injection once every 3 months, but most importantly, the treated eye of a person was compared to the contralateral eye that didn't receive treatment. In the phase 2/3 study, all these things changed. So the dose was only half of the initial dose. The injection frequency was only half, so once every 6 months. And instead of now looking at one eye versus the other eye, now the setup was defined in such a way that there was one person compared to an untreated person. So I think that these are the most likely explanations for the failure of the trial. the trial rather than that the drug didn't work. So luckily, as Camiel already pointed out, French company Sepal Bio took over these two programs, and the first patients are already being dosed or recruited. So I think that's very good. And in the last few minutes of my talk, I would like to share some data that we are currently doing on the ABCA4 gene. Again, discovery in the Radboud UMC and now being further followed up by our spin-off startup company Asterna. And ABCA4, some people in the audience may have heard about this gene and protein. It's an important protein in the visual cycle to clean up waste products of this phototransduction cascade. And it's a very genetically or allelic heterogeneous disease, because to date we know that there are more than 1,200 different variants that underlie Stargardt disease, which is the phenotype, the ocular phenotype that is associated with ABCA4 variants. So today I would highlight only two of the variants that we're working on. The first one is a very common variant, one of the most common variants in the Netherlands. And this variant is located in exon 6. Here this blue arrow points to the nucleotide that's mutated, and it changes into a T. And it's exactly at the boundary of the exon and the intron. And you can appreciate here the scores of the splicing programs. These are bioinformatic prediction tools. And you see that because of this G to T change, the strength of this splice site is going down. And a little bit down into the intron, there's another splice site predicted. This has a lower score than the original one, but it has a higher score than the mutant one. So you can imagine that instead of splicing here, the splicing machinery now decides to splice here because this splice site is a stronger splice site than this one. So we investigated that with a lot of people, are all mentioned here. We worked with retinal organoids derived from patients that are homozygous for this variant. And here you can nicely see that indeed the product in the patient is larger than the product in the isogenic control. And it's exactly this 35 nucleotides that are inserted into the transcript. Our approach using the antisense oligo was to design an oligo that would block the alternative splice site and hopefully forcing the splicing machinery to go back to the original site. So we did that. Again, we did a lot of work that I don't have the time to share, but we ended up with a very nice candidate, AUN number 38, and then the 21 nucleotide version. And here you see a nice dose-dependent effect of our oligo. The upper band is the wrong product, the lower band is the correct product. And you nicely see with an increasing dose of oligo that there's also an increase in correct transcript. We quantified that up to 60–65% of transcript is corrected. And we also checked whether that would lead to an increase of ABCA4 protein with a Western blot analysis. And again, here by this band here, you see that in the treated patient cells that normally don't have any ABCA4 protein at all of a sudden you get a nice correct expression of your ABCA4 protein. The last experiment that we did is to check where the oligo ends up if we inject that into the retina of a wild-type mouse. And here the red staining shows that the oligo is taking up very nicely, very broadly distributed in all retinal layers. This is 1 week after exposure, but we also have nice data that it will stay there until at least 3 months after one single injection. So the current steps we're currently taking is a rabbit tolerability study to see if our oligos are well accepted in the rabbit eye. That's the safety aspect. We're looking at starting GMP manufacturing, working towards the design of an IND-enabling safety study. And at the same point are also collecting natural history disease data and working on the right clinical endpoints to avoid the trial being not working effectively because of selecting the right or the wrong endpoints. Then the last minute, 1 or 2 extra slides on moving from rare to ultra rare, because the variants that I showed so far, they're present in at least a couple of thousand individuals worldwide. It's still rare, but it's substantial. But for instance, this variant here, it's present in only, up to date, we know 4 people that have this variant. And for one of them, we are working on an NS1 therapeutic intervention, similar to the KJ program that was shown earlier today. This variant also creates a pseudoexon, so we designed oligos to block that pseudoexon recognition. Here you see in this graph that for sure ASO3 and ASO4 are capable of correcting the pseudoexon containing transcript to the correct transcript. We quantify that here, so the higher the black bar, the better it is. And we also show that, especially for ASO number 3, with 5 and 10 micromolars, we not only get a rescue of the RNA, but we also get a significant increase of the ABCA4 protein. So we're now trying to develop this towards an NF1 treatment, hopefully to be started soon. So with that, I would like to end. For the commercially viable targets present in a couple of thousand patients, we want to develop that within our startup Asterna. And for these ultra rare cases, these NF1 studies, we work together with the Dutch Center for RNA Therapeutics, which is a national initiative to take care of this problem broadly, not only for retinal disease, but also for brain disorders, for muscle disorders, for skin disorders, with all different academic institutes in the Netherlands. And we also work together with the NS1 Collaborative, which basically does the same, but then more globally. They are located in the US, but they also support European research. So to take home, I think that ASO-based splicing modulation is a versatile approach. It's not working for all mutations, but for those that have a splicing defect, it's a very interesting and appealing therapeutic intervention approach. So far, they're well tolerated and also in individual cases demonstrate signs of efficacy, but it's very, very crucial to have a clear tailor-made design, I would almost say, for every clinical trial in order to not only show that the drug works, but also to make sure that it will get access to the market. And also moving from rare to ultra rare is possible via these alternative routes that we are now using and trying to use with the NS1 Collaborative. So these are the people that did all of the work, the bodies that fund my work, and I'm happy to take questions.
Well, thanks to you all for your presentations, for the nice discussion. We have to wrap up because it's 2 o'clock. I would like to thank Lucie Poitou for organizing with her team. And well, have a nice day, everybody, and a good weekend ahead.